Thank you, Carolina, for organizing this very interesting seminar series, and I really would like to invite everybody interested in Vesicle to participate and give talk. It's working extremely well. At least we are here from the United States, Australia, and New Zealand. We are now going to be able to have people from Europe participating. All right, so I'd like to introduce and be glad to introduce Dr. Shiv Ram Krishn. Shiv was born in India and obtained his biology degree and master degree in biochemistry from the Rajasthan University of Health Science in Jaipur. Shiv joined the graduate program at the University of Nebraska Medical Center in Omaha and joined the laboratory of Dr. Surinder Batra and obtained a PhD in biochemistry and molecular biology under the supervision of Dr. Batra. In his graduate research studies in Dr. Batra laboratory, he explored the utility of mucins as early stage markers for colon cancer and pancreatic cancer. In 2017, he remained in the solid tumor area, but decided to switch to prostate cancer since his interest in cell adhesion molecule really were, a good fit for my laboratory. And under Dr. Batra's recommendation, he decided to, he accepted to take a position with us as a postdoctoral research fellow. And he has been with us since early 2017. Shiv has done a wonderful job. He has already presented extracellular vesicle research at the Gordon Conference in 2018 and various national and international meetings. So I'm very glad they will today be able to present his new finding published in the Journal of Extracellular Vesicle this year in this seminar series. Shiv is also a member, he told me to remind everybody, he's a member of ISEV, the International Society of Extracellular Vesicles, which is a great society that sees Carolina here as a board member. And would like to encourage everybody to be part of the society because it has a lot of communication and spreads information around the globe, I would say, on extracellular vesicle research. Okay, thank you, Shiv. I'm glad you accepted to give a talk here in the seminar series today.
Hello, everyone. To begin with, I would like to thank Carolina to provide me this opportunity to present in this WebEVTalk seminar. And today I will be presenting our work on how αvβ6 integrin in cancer cell-derived small extracellular vesicles enhances angiogenesis. To begin with, I would like to give a brief background about prostate cancer. So instead of showing any improvement with regard to its new estimated cases or number of deaths due to prostate cancer, every year we are seeing a rising trend. Prostate cancer is the most common urologic malignancy in American men. And as we can see here, for 2020, it has been estimated that there will be approximately 191,930 new cases of prostate cancer, which accounts to 21% of all the cancer cases in men. It has been estimated that every 1 in 9 American men will get prostate cancer during his lifetime. Not only the prevalence, but also with regard to mortality, it is the second leading cause of cancer death in American men, with an estimated cases of approximately 33,330 deaths for 2020. So when we look towards the survival of prostate cancer patients, it has been shown that prostate cancer patients with localized or regional disease, they have a relative 5-year survival rate of up to 100%. But the problem comes in patients with metastatic prostate cancer who exhibit poor outcome with a relative 5-year survival rate of approximately 30%. And one of the major factors associated with regard to this metastatic prostate cancer disease and its poor outcome is the increase in blood vessel count during incidence of this metastatic disease. Not only that, with increase in prostate cancer histology grade, along with the increase in its Gleason score, the microvessel count increases. And therefore, it is very important to unravel those molecular mechanisms which are associated in aggressive high-risk prostate cancer and associated with angiogenesis in particular. So we wanted to know that what are those factors which are responsible for increased angiogenesis in metastatic prostate cancer disease. So there is a growing precedent and a lot of exploration going on these days with regard to utility of small extracellular vesicles or role of small extracellular vesicles in modulating the prostate tumor microenvironment, which also includes its role in increasing endothelial cell proliferation along with tubule formation. So these small extracellular vesicles play a multitude of role in prostate tumor microenvironment. As we can see on this cartoon, they also do immune modulation by presenting antigens to dendritic cells or by differentiating myeloid cells to MDSCs, which can inhibit T cell infiltration. Myeloid cells can also be differentiated to anti-inflammatory M2 macrophages, which in turn secrete more of VEGF or IL-6 that are known to be pro-angiogenic molecules, thereby increasing the incidence of angiogenesis. Other than this, there are published reports which suggest that prostate cancer cell-derived exosomes are responsible for extracellular matrix remodeling that leads to increased metastasis. Also, prostate cancer exosomes play a major role in chemoresistance, motility, metastasis, and changing the glycolytic landscape of the prostate tumor microenvironment. Not only this, indirectly, these prostate cancer exosomes impact the stromal cells in the tumor microenvironment, also the stem cells in a tumor microenvironment, increasing their proliferation, which in that case secrete more of VEGF or urokinase plasminogen activator or growth factors like FGF, and ultimately lead to increased endothelial cell proliferation and tubule formation. So one of the major factors which is responsible for prostate tumor microenvironment increased angiogenesis and thus in prostate metastasis are these small extracellular vesicles. So with regard to these vesicles, when we look at this cartoon, these vesicles can be differentiated into multiple different subtypes based on their biogenesis or based on their size or based on their function or the cargo that they carry. As we can see here, that there are EV subtypes which are designated as exomere, which are approximately 35 nanometer to 50 nanometer in size, or small extracellular vesicles which can be further subtyped into small exosomes that range from 60 to 80 nanometer, or large exosomes that range from 90 to 120 nanometer, and small extracellular vesicles that are up to 200 nanometer in size. Along with it, there are other subtypes of large extracellular vesicles which are designated as either microvesicle, which are which range from 100 to 1000 nanometer, exophores, microsomes, and large oncosomes, which are considered to be 1 to 10 micron in size. With regard to when we look at biogenesis of these extracellular vesicles, although biogenesis of the small extracellular vesicles that range from 60 to 200 nanometer and microvesicles that range from 100 to 1000 nanometer is very well defined, but biogenesis of other subtypes like exomeres and large oncosomes has not been studied a lot. With regard to cargo, these vesicles carry all sort of cargo, which include RNA, DNA, proteins coming from plasma membrane or from cytoplasm, mitochondria, Golgi bodies, endoplasmic reticulum, or nucleus. So since extracellular vesicles comprise of different subpopulations of different size vesicles, in this presentation and our study focuses on small extracellular vesicles ranging from 50 to 200 nanometer. And this brings me to alpha V beta 6 integrin, the molecule that I am working on, which is relevant to prostate cancer, small extracellular vesicles, as well as angiogenesis. So I'll briefly introduce alpha V beta 6 integrin. It is a heterodimeric cell surface protein where the beta 6 subunit interacts only with alpha V subunit. It is absent in normal prostate, as we can see here. In normal prostate, there is no expression of alpha V beta 6 integrin, while it is highly upregulated in prostate adenocarcinoma. As we can see here, in different prostate adenocarcinoma tissues, its expression is very high. Also, its expression is very high in bone metastasis, as we can see here. Alpha V beta 6 integrin is known to bind extracellular matrix proteins like fibronectin, and there are very well it's very well known not only in prostate cancer but also in many other different cancers that it promotes tumor cell growth, adhesion, migration, and invasion. Our group recently published that alpha-v-beta-6 integrin is actively packaged in small extracellular vesicles, as we can see here in this electron microscopy image showing small extracellular vesicles derived from PC3 prostate cancer cells, and through sucrose density gradient centrifugation, we show that alpha-v-beta-6 integrin is actively packaged in small extracellular vesicles along with extracellular vesicle markers such as CD81 and flotulin 1. Not only that, it is actively packaged into extracellular vesicles, but it is also transferred via these SCVs from donor cells to alpha-v-beta-6 negative recipient cells, as we can see in this Western blot shown here. And once it is transferred to the alpha-v-beta-6 negative recipient cells, it localizes to the cell surface. In another study published by our group, we have also shown that alpha-v-beta-6 integrin shows in small extracellular vesicles shows a crosstalk with monocytes. Alpha-v-beta-6 integrin expressed in small extracellular vesicles is transferred to the monocytes, leading to their M2 polarization. Whereas upon downregulation of alpha-v-beta-6 integrin in small extracellular vesicles, the M2 polarization of monocytes is inhibited. It is very well known that M2 polarization of monocytes leads to increased prostate cancer tumor growth, while M1 monocyte inhibits this cancer growth. So based on this study, it can be suggested that inhibition of alpha-v-beta-6 integrin can be a very good therapeutic approach, immune-based therapeutic approach to inhibit the prostate tumor growth. With regard to angiogenesis, not much has been studied about alpha-v-beta-6 integrin. Alpha-v-beta-6 integrin is known to play a role in remodeling of vasculature. There are few studies which suggest that this integrin is expressed in small subset of blood vessels during later stages of a healing wound. As we can see in this picture, this is a picture of a healing wound where alpha-v-beta-6 integrin is expressed in the epithelium, whereas a blood vessel near it also start expressing alpha-v-beta-6 integrin. So blood vessels are basically known not to express this integrin, but during conditions of a healing wound, there is induction in expression of this integrin. Along with this, it has also been shown that CMV infection induces integrin alpha-v-beta-6 expression in endothelial cells, which leads to activation of downstream signaling and many other effector functions. So based on all these previous studies, we hypothesized that small extracellular vesicle associated alpha-v-beta-6 integrin shed by prostate cancer cells impacts the angiogenic potential of endothelial cells in the prostate tumor microenvironment. And our study, for the first time, investigate the endothelial cell modulatory role of alpha-v-beta-6 integrin in iodexonal density gradient purified vesicles that are devoid of any contaminants. So to test our hypothesis, we first characterized the prostate cancer cell-derived SCVs for expression of alpha-v-beta-6 integrin and SCV markers. So we isolated SCVs from PC3 cells and we characterized it by nanoparticle tracking analysis as well as zeta view. As we can see here in figure A and figure B, our SCVs derived from PC3 cells, most of the SCVs derived from PC3 cells are less than 150 nanometer in size. We then characterize these SCVs by Western blotting. As we can see here, in comparison to total cell lysate in SCVs, beta-6 integrin is enriched along with extracellular markers such as tetraspanins such as CD63 and CD81, whereas expression of endoplasmic reticulum marker calnexin is absent in these SCVs. We further purified these vesicles via iodexonal density gradient centrifugation and then did analysis for expression of beta-6 integrin in different fractions derived from these iodexonal density gradient centrifugation. And what we observed is that beta-6 is maximally expressed in a fraction corresponding to 1.123 gram per ml, along with SCV markers such as CD63, CD81, and CD9, whereas endoplasmic reticulum marker calnexin is absent in all these fractions. We next wanted to know whether prostate cancer cell-derived SCVs efficiently transfer αvβ6 integrin to endothelial cells. For this, we utilized human microvascular endothelial cell called HMEC1. We incubated HMEC1 with PBS or with PC3 derived SCVs for different time points, 2 hour, 4 hour, 8 hour, 16 hour, 24 hour. And then we performed qPCR to look for beta-6 mRNA expression. We found that there was no induction of beta-6 mRNA in HMEC1 upon incubation with PC3 small extracellular vesicles. This led us to investigate whether beta-6 integrin is being transferred to these endothelial cells as an active protein. So because beta-6 integrin is a cell surface receptor, we performed FACS analysis to look for cell surface expression of beta-6 integrin on HMEC1 cells after incubation with beta-6 expressing SCVs. What we found is that beta-6 is not only transferred to HMEC1, but it also localizes to cell surface. We further confirmed our cell surface expression of beta-6 integrin in HMEC1 cells by Western blotting. For this, we incubated HMEC1 with beta-6 expressing SCVs for different time points, 6 hour and 24 hour. As we can see here, a very low level of beta-6 integrin was transferred to endothelial cells in 6 hours, while a very robust amount was transferred in 24 hours. In order to rule out that whatever we are looking here in Western blot is due to any vesicles sticking on cell, we also did acid wash, and we found that there was an efficient transfer of beta-6 integrin in HMEC1 cells. In order to further functionally characterize role of beta-6 integrin transfer via SCVs to endothelial cells, we utilized PC3 cells stably transfected with SH control shRNA or shRNA targeting beta-6 integrin subunit, or shRNA targeting beta-5, another alpha V binding integrin, that is beta-5 subunit. We isolated SCVs from these stably transfected cells and characterized them first by nanoparticle tracking analysis. As we can see here, most of these vesicles from PC3 SH control cell or cells having knockdown of beta-6 or knockdown of beta-5 integrin, the SCV size is less than 150 nanometer. We then characterize these SCVs by Western blotting. As we can see here, beta-6 integrin subunit is expressed in SH beta-5 as well as SH control cells, while its expression is significantly reduced in SCVs derived from cells where shRNA to beta-6 was transfected. Along with this, we found that SCV markers are enriched in all the SCVs in comparison to total cell lysate, and presence or absence of beta-6 did not alter the levels of these SCV markers, CD63, CD81, or TSG101. We then utilized these SCVs to incubate with endothelial cells, and we performed tube formation assay, in vitro tube formation assay. In our in vitro tube formation assay, we found that beta-6 upon incubation with beta-6 expressing SCVs, there was a significant increase in number of nodes, junctions, and tubules in comparison to PBS treated control, whereas SCVs lacking alpha V beta-6 inhibit formation of nodes, junction, and tubule in comparison to PBS treated control as well as SH control. We also generated cell line where we stably transfected empty vector as well as beta-6 cDNA expression vector. C4-2b cell line do not endogenously express beta-6, therefore we utilized beta-6 cDNA expression vector. We isolated SCVs from these stably transfected cells, characterized them by nanoparticle tracking analysis. As we can see here, most of the SCVs derived from these cells are less than 150 nanometer size. We also characterize these SCVs by Western blotting. The exogenously expressed beta-6 integrin is sorted into SCVs, as we can see here, while endoplasmic reticulum marker is absent in these SCVs. Also, similar to our knockdown cells, there was no impact on expression of SCV markers such as ELIX, TSG101, and CD9. upon expression of αvβ6 integrin in C4-2b cells. We utilize these SAVs to further assess impact on HMEC1 angiogenic potential. And as we can see here, the β6 expressing SAVs significantly increase the number of nodes, junctions, and tubules in endothelial cells in comparison to previous treated control or mock vector-derived SAV control. Further, we wanted to know whether this increased tube formation is because of increased proliferation of these cells. In order to find this, we performed trypan blue dye exclusion assay. We incubated HMEC1 cells with PBS or SEVs derived from C4-2b cells, mock or αvβ6 transfected cells. And we found in our assay that there was no significant difference with regard to cell number, suggesting that there is no major impact on proliferation of endothelial cells upon incubation with these SCVs. Next question that we asked was whether there is any significant impact on motility of these endothelial cells upon incubation with prostate cancer cell-derived SCVs that express αvβ6 integrin. As we can see here, upon incubation with SCVs that express αvβ6 integrin, there was a significant increase in motility of endothelial cells, whereas upon incubation with SCVs lacking αvβ6 integrin, there was a significant decrease in motility of these cells. Similarly, with C4-2b αvβ6 SCV derived SCVs, there was a significant increase in motility of endothelial cells in comparison to previous treated control as well as mock cell-derived SCVs. Since there is an overlap with regard to size in large extracellular vesicle, which are in range of 100 to 1000 nanometer, as well as in small extracellular vesicles, which are in range from up to 200 nanometer, 50 to 200 nanometer. We also investigated whether large extracellular vesicles isolated by 10,000 G centrifugation express αvβ6 integrin. We purified those 10,000 G vesicles by iodoxanol density gradient, and by Western blotting, we investigated whether different fractions express β6 integrin. What we found is that β6 integrin is in fact expressed in these large extracellular vesicles and in fractions corresponding to in fractions of SCVs corresponding to different densities, along with SCV marker CD tetraspan in CD63 and CD81. We next asked whether beta-6 expressed in these large EVs can impact tube formation of endothelial cells. For this, we took two different fractions, fraction 4 and fraction 5 of LEVs, and we incubated with HMEC1, and we found that there was no significant difference with regard to formation of nodes, junctions, and tubules by endothelial cells. Whereas as a control, when we utilize fraction 3 from αvβ6 expressing small extracellular vesicle, there was a significant increase in formation of nodes, junctions, and tubules. So this brings me to the interim summary, where we found that αvβ6 integrin protein is efficiently transferred by prostate cancer cell-derived SCVs to endothelial cells, and it localizes to the cell membrane. Further, αvβ6 integrin is expressed in prostate cancer cell-derived SCVs, significantly increases the tube formation by endothelial cells. And not only this, αvβ6 integrin expressed in prostate cancer cell-derived SCVs also significantly increases the motility of endothelial cells. This prompted us to ask a question that what is the mechanistic basis of angiogenic modulation of endothelial cells upon incubation with αvβ6 integrin positive prostate cancer SCVs. So our group have previously published that downregulation of αvβ6 integrin causes a significant increase in donor prostate cancer cells and their SCVs of signal transducer and activator of transcription 1, that is STAT1. Also, STAT1 is generally considered a tumor suppressor, but there is growing evidence that it can also act as a tumor promoter. Further, we found from literature that STAT1 is known to negatively regulate the angiogenesis in endothelial cells. Based on these studies, we asked the question that whether transfer of alpha v beta 6 integrin via prostate cancer SCVs impact STAT1, which is an inhibitor of angiogenesis in endothelial cells. To answer our question, we generated CRISPR-Cas9 mediated beta 6 knockout PC3 cells, and we first characterized SCVs derived from these cells. As we can see here, wild type cells are the cells which express beta 6 integrin, whereas two different clones, knockout 5 and knockout 7, do not express beta 6 integrin in total cell lysate as well as in SCVs derived from them. But we found that as we previously showed with knockdown cells or with overexpression of alpha v beta 6 integrin, there was no impact on expression of SCV markers. Similarly, upon CRISPR-Cas9 mediated knockout of beta 6 integrin subunit, we didn't observe any difference in level of CD63, CD81, TSG101, or CD9 in SCVs derived from beta 6 knockout cells. We utilized these SCVs to incubate with HMEC1 endothelial cell, and we found that beta 6 integrin is efficiently transferred via wild type SCVs, whereas upon incubation with knockout 5 or knockout 7 cell-derived SCVs, we didn't see any expression of beta 6 integrin. We further investigated whether incubation with SCVs that lack alpha v beta 6 integrin, does it change expression of phosphoSTAT1 tyrosine 701, which is considered to be an inhibitor of angiogenesis. And we found that in endothelial cells, upon incubation with beta 6 integrin lacking SCVs, there was a significantly increased expression of phosphoSTAT1 tyrosine 701, whereas STAT1 levels, total STAT1 levels, do not change in these cells. We also corroborated our findings with SCVs derived from beta 6 knockdown or beta 5 knockdown cells, and we found that upon incubation of endothelial cells with alpha v beta 6 lacking SCVs, there was a significant increase in phosphoSTAT1 levels in both with both PC3 as well as C4-2b cell line derived SCVs. We also found that there is an antagonistic effect between STAT1 and survivin in gastric cancer cells and tissues. Also, we have previously published in our lab, from our lab, that expression of alpha v beta 6 integrin in prostate cancer cells results in an androgen receptor mediated increased expression of survivin. Survivin is a molecule that is known to be upregulated in most, if not all, cancers, and it is considered as a potential oncotherapeutic target. It is essential for cell division and can inhibit cell death. And also, from literature, we found that survivin is a critical mediator of angiogenesis. So all these studies prompted us to ask a question whether transfer of alpha v beta 6 integrin via prostate cancer SCVs impact survivin levels, which is a pro-angiogenic molecule in endothelial cells. So in order to answer this question, we first wanted to know whether survivin is expressed in prostate cancer cell-derived SCVs. For this, we purified SCVs derived from PC3 cells by iodexonol density gradient centrifugation, and by Western blotting, we investigated whether survivin is expressed in low density small extracellular vesicles. And what we found is that survivin was expressed in different fractions from iodexonol density gradient, along with SCV markers such as ELIX and TSG101 and tetraspanning such as CD63, CD81. We further asked the question whether survivin levels are impacted by knockdown of beta 6 integrin in prostate cancer cells, PC3 prostate cancer cells. What we found is that in total cell lysate, there was no change in level of survivin, whereas upon knockdown of beta 6, survivin levels were significantly reduced in small extracellular vesicles derived from these cells. We incubated these SCVs with endothelial cells, and we wanted to know whether there is a change in level of survivin in HMEC1 endothelial cells. What we observed is that upon incubation with β6 integrin lacking SCVs, there was a significant reduction in level of survivin in endothelial cells. So since we hypothesized that alpha V beta-6 integrin can be transferred via prostate cancer cell-derived SCVs to different other cells and specifically endothelial cells, we wanted to investigate whether alpha V beta-6 integrin is expressed in blood vessels of human prostate cancer tissues. For this, we conducted immunohistochemistry on prostate and serial section of prostate cancer tissues, along with to find out the level of alpha V beta-6 integrin in these tissues, along with von Willebrand factor, which is expressed in blood vessels. As you can see here, the red arrowheads show expression of von Willebrand factor in blood vessels that express in blood vessels, whereas yellow arrowheads show simultaneous expression of alpha V beta-6 with VWF, and green arrowheads show expression of alpha V beta-6 integrin in prostate cancer cells. So what we observed is that in fact, alpha V beta-6 integrin is expressed in blood vessels along with blood vessel markers. And this observation was specifically observed in high Gleason score prostate cancer tissues. So this brings me to overall model of our study, where we found out that SEV mediated transfer of alpha V beta-6 integrin from prostate cancer cells to endothelial cells promotes angiogenesis. What we found is that from prostate cancer cells derived SEVs, they express alpha V beta-6 integrin. These SEVs are transferred to endothelial cells where alpha V beta-6 integrin is localized to the plasma membrane. Not only it localizes to plasma membrane, but functionally it impacts the tube formation. It increases the formation of nodes, junctions, and tubules. Also, motility of endothelial cells is increased significantly. Mechanistically, we observed that upon transfer of alpha V beta-6 integrin to endothelial cells, there was a significant reduction in level of phosphostatin tyrosine 701, whereas there was significant induction or increased level of pro-angiogenic molecule survivin. Therefore, owing to our these novel findings, we speculate that targeting alpha V beta-6 integrin in combination with current anti-angiogenic therapies may provide a novel approach to develop effective therapies against prostate cancer. This brings me to the acknowledgement slide. I would like to thank Dr. Languino for her mentorship, all our lab members for their support and help with regard to conducting this study in different capacities, and all our collaborators from Sidney Kimmel Cancer Center as well as from different institutes, Vista Institute and Yale University. Thank you very much, and I'm happy to take any questions. Thank you.
Thank you. Thank you, Shiv. Carolina asked me to run the Q&A session, and then Carolina will step in. Thank you, Shiv. So I see there are questions already on the chat here. The first one is from Navid. I think it's you. You were the first. Oh, where is it? Hold on. No, Christopher. Sorry, Chris. Chris, are you here? Yeah, you can unmute yourself. Okay, good.
Hey, nice job, Shiv. That was really thorough. Lots of data, really good data. So yeah, because you mentioned kind of in the introduction, just in the context of alpha V beta-6, that like there was like immune cells that responded to it. I was just wondering if there's any like evidence for like selectivity of these EVs with the beta-6 integrin, and just other stromal factors, because it is like this adhesion molecule.
Yeah, I mean, it's a very good question. Thank you, Chris. So what I see it is that these EVs are coming from prostate cancer cells in prostate tumor microenvironment. They express alpha V beta-6 integrin. Now they are transferred to, like as we have published, that they are transferred to other prostate cancer cells. They are transferred to monocytes. And then, as you mentioned, that is it that they have any sort of selectivity towards stromal cells, which I believe that it can be, because these stromal cells might be expressing a lot of fibronectin, and alpha V beta-6 integrin binds to fibronectin. So there can be, but we haven't investigated whether it is selective when we, if we keep endothelial cell and stromal cell together, whether alpha v beta 6 integrin expressing SCVs will selectively go and bind to stromal cells, or they will have any preference to bind to endothelial cells, go and uptaken by endothelial cells. But it's a very good question. Yeah.
Thank you, Chris. The next question is from Dr. Kuckerman. Eti, are you there?
Yes, I'm here. Can you hear me?
Oh, yes, definitely. Yeah.
All right. So great talk. I wanted to know whether in a similar experiment to the ones that you did with the SCVs generating angiogenesis, whether you were able to use a simple anti-alpha v beta 6 antibody that it will be blocking the function, in that case, inhibit the angiogenesis.
Yeah, and thank you, Dr. Kuckerman. In our lab, we have previously utilized antibodies inhibiting alpha v beta 6 in mouse models, where we found that when we inject these antibodies subcutaneous and in mouse model of prostate cancer, there was no development of tumor. In this study, we did not block SCVs with alpha v beta 6 antibody like 6-3G9 because we utilized these cells where beta 6 integrin was already inhibited, like inhibition via knockdown, or its knockout via CRISPR-Cas9, or we utilized a cell line where we overexpressed beta 6 integrin while endogenously it was not there. And we could clearly see that there was a significant impact on angiogenesis. But yes, we did not utilize 6-3G9 antibody, which inhibits alpha v beta 6, to block SCV effects on endothelial cells specifically. Yeah.
And is there any angiogenesis defect in the animals that previously were treated with the antibody, or in animals that are knocked out for beta 6? Is there any report of angiogenesis defects?
Yeah. So the major impact that we saw was that there was no tumor growth. Either it was very, very small tumor, or there was no tumor growth. So we lacked those resources where we could investigate whether what was happening with regard to blood vessel formation in those tumors. So unfortunately, I won't be able to answer that question unless we do that kind of experiment again.
Thank you so much.
Thank you, Eti. The next question is from Alison Shoop. Alison, are you here? I see you.
Yep. Hi, yes, I am. Hi, Shiv, great talk. My question was, if you think the angiogenic response that you're seeing is prostate cancer specific, so do you know what the expression of beta 6 is like in maybe other cancers or even non-cancerous cells? Like, could this just be an effect of having beta 6 in the EVs?
Yeah, so with regard to impact of alpha v beta 6 integrin on angiogenesis in cancer, this is the first study which shows that beta 6 integrin can have some impact on angiogenic potential of endothelial cells. No previous study specifically in cancer has shown it. Beta 6 integrin is known to be upregulated in many different cancers like breast cancer, colorectal cancer, but none of these studies have previously studied role of alpha v beta 6 integrin or role of alpha v beta 6 integrin in small extracellular vesicles to induce any angiogenic potential.
Cool, thank you.
Thank you, Alison. The next question is Antula. Antula, are you here? Yes, I see you. Antula is here.
Yes,
hi, Antula.
Thank you. Hi, can you hear me?
Yes.
Sure. Yes.
Okay. Thank you for a very nice talk. Very interesting. When you use your EVs that are depleted of the integrins and you inhibit angiogenesis, or the cells are not stimulated to grow, so are they senescing? For example, in an in vivo situation, would these vessels now become mature, for example, and not respond to angiogenesis, which may not be amenable to anti-angiogenic therapies anymore? So you're actually directing the cells, the endothelial cells, into a more mature vessel formation.
Yeah, sure. So what we observed was we first performed the MTT assay after incubation with these SCVs, which were coming from alpha V beta 6 expressing cells or alpha V beta 6 lacking cells. And what we observed was that in MTT assay, we were seeing a difference with regard to their viability. Although those results were not significant, but it was giving us an idea that there was something happening with regard to mitochondrial activity in those cells. But when we try to more specifically answer our question that whether it is impacting proliferation, we then conducted this trypan blue dye exclusion assay to know whether these cells are actually proliferating. So what, as you say, that are they not growing? No, that's not true. They are growing because when we incubate them with beta 6 expressing SCVs, we get to see more tube formation. We get to see more nodes and junction formation and more motility. But in fact, they are not, I will say that cell number was not statistically significant in terms of difference with regard to beta 6 expressing SCVs or lacking SCVs. We didn't investigate whether there was any senescence going on in endothelial cells. We didn't look for that.
Thank you.
Mm-hmm.
Yeah. Thank you, Antula. And then there is a question by Navneet. Navneet, would you like to ask a question? You're muted. You're muted.
Hi, Shiv. Hi, Lucia. Great talk.
Hi, Navneet. How are you?
So my question is about, you showed in one of the slides, and I kind of missed it. Was there alpha V beta 6 mRNA in the vesicles? And what about that or other RNA's contribution for angiogenesis?
Yeah.
And,
so
yeah, continue.
Yeah, Navneet, it's a great question. I, in my hands, did not investigate whether beta 6 integrin mRNA is present in these SCVs, but our published paper from our group have previously shown that beta 6 mRNA is absent in these SCVs. Also, when we incubate endothelial cells or other prostate cancer cell types with SCVs coming from beta 6 expressing cell or beta 6 knockdown cells, we don't get to see any induction of beta 6 integrin in recipient cell as well. So we think that there is no beta 6 integrin mRNA being transferred via these SCVs.
Mm-hmm. And the additional question was that if it's just alpha V beta 6, and if we encapsulate or transmit, if it's on the surface of liposomes only, right? There's no other factor that has an effect. Have you thought of just making exosomes with the protein of interest and then inducing angiogenesis?
If liposomes can efficiently go inside the cell and transfer alpha V beta 6 as an active molecule, and if it gets translocated to the plasma membrane, I definitely believe that it will have impact on angiogenesis, because ultimately it's transfer of this protein that we are claiming having impact on angiogenic potential. So if liposomes carry that potential to go inside the cell and then transfer this protein efficiently without getting degraded in lysosomes or those factors, then definitely there should be some impact on angiogenesis.
Okay, thanks. Just last comment.
Thank you, Navneet. Yeah, please go ahead. Yeah, sure.
Your Western blots are beautiful.
Oh,
and I could only imagine how much media you have cultured and then use the
Credit goes to Dr. Languino.
Thank you, Navneet. Okay, then I have Dr. Kazuki Sugahara. Kazuki, yeah, he has a few questions.
Hi, Shiv. Thanks for a great talk. I have two questions, basically. One is that I could see that, the cells that received alpha V beta 6 did a lot of things like angiogenesis, etc. But I'm wondering how well those integrins are incorporated into the system. Like a simple way to show it would be to attach those integrins to their substrate and see whether it induces the conventional signaling pathway. I'm wondering whether you saw any of those.
Yeah, it's a great question. I think we can definitely try to adhere those cells on fibronectin or vitronectin, and upon incubation with SCVs, we can see whether when alpha v beta 6 integrin is transferred to plasma membrane, does it form focal contacts, and does downstream signaling mediated through FAC or SERC, which majority of integrins are believed to conduct, although we didn't look into this yet. Yeah.
Okay. The other question, or it's more of a comment, I think the million-dollar question is whether this is happening in vivo. You know, you showed that last slide where you saw alpha v beta 6 integrin in the blood vessels in human specimen.
Yes, specifically in high Gleason grade specimens.
Right.
Yeah.
But of course, we don't really know whether it was a result of exosome transfer, whether it was because of other stimuli. One interesting experiment would be to grow your human tumors in nude mice and see whether you can detect human alpha v beta 6 in the blood vessels there, because the blood vessels are coming from the mice. So if you see human protein there, then you sort of have a good evidence or a very good, you know, stuff that you can say that maybe it's really happening in vivo. Just a comment.
No, very, 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 yeah, very.
Yeah, and
No, go ahead.
thank
You
you
go,
for suggesting this question. Thank you for suggesting this question. But the only thing is that even if we do that we put human tumors and then we look for mice blood vessels, it doesn't tell us for sure that it was the transfer in blood vessels was mediated only by extracellular vesicles, small extracellular vesicles. Because there are reports which suggest that beta 6 integrin is also found in serum of patients, certain patients. So there might be certain ways where it can come extracellularly other than extracellular vesicles. But definitely it's a great experiment. Yeah.
Yes, thank you, Kazuki. No, we have been discussing so much about a way to prove in vivo what the system is, but you're right, that needs to be done. That's an important point. Yeah, thank you. I have one more question from Derek. Derek, are you here? Yeah. Okay,
yep,
Derek is here.
I'm here.
Hi, yep.
Hey, Shiv, great talk. So I was just wondering if you thought that the increased αvβ6 expression alters the ability of the circulating tumor cells. So if the prostate cancer cells make their way into circulation, if it would alter their ability to extravasate from the circulation, and is this helping to maybe form a pre-metastatic niche or anything like that? Do you think it has any role in that? And then do you think if you knock out the cells, would that alter the ability, of the cells to kind of get out of circulation?
Yeah. Hi, Derek. Great question. So I am not aware of whether alpha v beta 6 integrin is expressed in circulating tumor cells coming from prostate cancer tumor. But one thing is pretty sure that when these EVs are coming out of prostate tumor, they might definitely form metastatic niche at distant sites, maybe at lung, because lung is known to express alpha v beta 6 integrin. And we are currently trying to investigate that, specifically in human patient samples. We are trying to find out. So yeah, this is ongoing work.
Okay, thanks.
Yeah, so I don't see other question in the chat. If there are other questions, otherwise I will simply ask Shiv, can you comment on the large extracellular vesicles versus small extracellular vesicles? I mean, they both have beta 6, you told us, right? We haven't had the chance to talk about this much. What do you think is happening?
Yeah, so this point came to our mind that we wanted to investigate beta 6 integrin in large extracellular vesicles and small extracellular vesicles because their size overlap to some extent between 100 to 200 nanometer. So we investigated expression of beta 6 integrin and we found that beta 6 integrin is present in large extracellular vesicles. Now, the major question was whether large extracellular vesicles which are expressing beta 6 integrin, can they impact the angiogenic potential of endothelial cells? But what we found was that there was no impact on angiogenic potential. So it kind of make us think about it that if these vesicles are also expressing beta 6 integrin, then why they are not inducing angiogenic potential? And only plausible reason that we could find is that large extracellular vesicles are not transferring active β6 integrin in endothelial cells, although we haven't investigated that yet. So that was one thing that we could think of.
To work on. Very good. Perfect. Then. Okay, thank you, Shiv. So I'm gonna, if there are no more questions, no. Okay, Carolina, to you. Okay.
Thank you so much, Lucia and Shiv. Very wonderful talk and very much details and experiments done very neatly. I really love it. Thank you so much for sharing it with us, and went through all the data and explaining to us all that beautiful work that you've done.